So it's my pleasure to kick off the first meeting of this year's Wednesday evening at the Genome Public Lectures. We're all sad that we can't do this in person this year, but thanks in part to Lea, as she'll tell you, we're hopeful that that might begin again in the following years. So Lea Starita is now an assistant professor in the Department of Genome Sciences and also the co-director of the Brotman-Beatty Institute Advanced Technology Lab. Before that, she got her PhD from Harvard Medical School and has been at the University of Washington for several years, first as a fellow with Stan Fields and Jay Shinduri before she started her own position. So Lea's done a lot of important work related to, say, BRCA cancer susceptibility genes, but today she's going to tell you about her work in the early days of the COVID-19 pandemic, where she was able to identify some of the first community spread of COVID, thanks to her previous work on the Seattle flu study. So before I spoil any more of it, I'll turn it over to Lea.
Okay, so thank you for inviting me to talk about our experience this year. It's been a kind of a wild ride, and I hope to take you along with me to kind of learn what happened in the last year and try to figure out how to make it not happen again. I've always really been interested in science. And this is a picture, dating from, my parents' hideous furniture, that this was about 8th grade and the unfortunate turtleneck. So I've always really been interested in science, and I majored in molecular biology in college. And then, as Kelly just told you, I went to Harvard Medical School for grad school. They have all of these beautiful kind of stately buildings. I, of course, was not in this building. Oops, where did it go? I was in this building behind it, which is appropriately called the Thorne Building. I learned a lot of biochemistry and skills at Harvard, and then I had wanted to learn kind of high throughput genomics, and that brought me to genome sciences. And my specialty is really genomic technology development. And so if there is a piece of DNA or RNA and you want it detected or sequenced, I am your girl, which is kind of what led me to the Seattle flu study. So the Seattle flu study had a very lofty goal when it was launched in 2018, a mission of transforming how infectious disease outbreaks were detected, how they were monitored, and then how they were also controlled. And since there was not a pandemic in 2018, the idea was to use the annual emergence and spread of seasonal flu as a model. And that's kind of hence the name, right? Also, everybody in the winter kind of thinks they have the flu, even if they don't. And another reason for the name is we kind of thought the next pandemic would be influenza. And so we called it the Seattle Flu Study. It was a nice, easy name. And this vision required a lot of innovation from, participant recruitment, community buy-in to get samples, high throughput testing and sequencing, and, a lot of informatics plumbing. And so to do that, this project wound up being based here at the University of Washington at the Brotman-Beatty Institute with faculty from University of Washington, UW-Medicine, Fred Hutch, Seattle Children's, and the Institute for Disease Modeling, which is now part of the Gates Foundation. So the first thing that we wanted to do, right, so to really build a respiratory pathogen observatory, you have to deeply sample the population. And to do this for a city the size of Seattle, it was going to take a lot of, we're going to have to get these samples from all sorts of different places. And to do this, this was led by our clinical team. So on the, this is Helen Chu on the left. She's a infectious disease specialist from the University of Washington and also the Washingtonian of the Year. And then there's Jan England, who knows everything about respiratory disease in children, and Michael Book from the Fred Hutch, who knows everything about respiratory disease in the immunocompromised, which are kind of a special class of people when you're talking about infection. So the first way we were going to get samples was the easiest, which is which took more negotiation than you would think, and that's kind of medical dumpster diving, right? So taking the remnant or leftover samples from hospitals. So sometimes hospitals will, will test somebody for flu, but the rest of the sample, they're just going to throw that out. And so we asked them not to throw it out, and then they charge us $10 and they give it to us, which is very nice. And then the second thing we can do, and we can get all the relevant information from, medical records, which is really, really handy. But we wanted to actively engage the community because the people who show up at the hospital or even just show up at the doctor are generally the people who are very sick, right? And so we wanted to figure out what was going on in the community, right? So these are kids who, probably just stayed home from school or, just kind of felt kind of bad. But so what we did was we built kiosks up around the city. So in high traffic areas. So for example, one of our best ones was at University of Washington's Student Center. And then at the library, we had them downtown at the Westlake Center. We were trying to get travelers, although trying to get somebody to stop at the airport was kind of difficult. But we had these things at, the light rail stations and all that sort of other stuff. But the biggest innovation was a program called Swab and Send. And so I'll tell you a little bit more about that, but if you, some of you folks from Seattle might remember that there were ads kind of around the city in the winter of 2019-20 that said things like this, your family can help fight the flu and here's how. And the way this worked was if you had respiratory infection, symptoms of an acute respiratory infection like cough or sore throat or headache, you could hit the seattleflu.org website. And we would actually send you a kit to use at home to swab your own nose. So you would swab your nose, put the swab in this transport media, package this back up, and actually just send it in the mail back to us. And it would come back to our lab at the Bratman Beatty Institute. And we wound up actually through local awareness and through kind of targeted social media advertising, we wound up doing 3,000 or collecting 3,000 of these types of samples even before the pandemic. And so when they get back to the lab, we want to know what's in them, right? So what do these people actually have? And where did it come from, right? So the first thing you do with these samples is you have to get the genomes of the viruses out, viruses or bacteria out. So we extract that RNA or DNA. And then we try to detect any of 27 different pathogens, which range from very common things like rhinovirus down to things that are way less common, like enterovirus D68 and other types of pathogens that are kind of emerging. And then we also, when we found a genome or when we found a pathogen, we were interested in sequencing that would move on to the sequencing platform. And this is Debbie Nickerson. So it turns out we would wind up dealing with 23,000 or so specimens in the first 10 months. And so we wound up we needed the big guns, and Debbie runs the Northwest Genome Center right here at Genome Sciences. And so she really gave us the firepower to be able to do this. But I want to do the first of a few little digressions on different little molecular biology how-tos here, right? So what we're looking at here is a chip that each one of these little holes holds a 33 nanoliter qPCR reaction, which is going to detect the pathogens. And I just want to tell you a little bit more about how that works. So PCR, and I stole this actually from Khan Academy, is a way to copy DNA. It stands for polymerase chain reaction. And if you have a little piece of DNA that you're going to target, so in this case, it would be in the genome of a bacteria or virus, you can rip these two strands of DNA apart. And by adding these little primers, so little pieces of DNA that bind to the template or the DNA you're looking for. You do that, and then the next thing that happens is that you add a polymerase, and the polymerase extends these primers and copies this original piece of DNA, right? And you cycle this, so it's a chain reaction. So you cycle this, and you get, from cycle 1, you get 1 copy, from cycle 2 is 4, and you exponentially expand the piece of DNA that you're looking for. And the Q, so the qPCR in all of our detection assays and basically all of the COVID tests that you guys have either taken or heard about, we add a probe to be able to detect this amplification in real time, right? So as the DNA is making copies and copies and copies, we want to be able to see that. And the way we see that is we add this thing called a probe. So it's sequence specific, it binds to that piece of DNA, and it has a fluorescent molecule on it that doesn't fluoresce if it's next to this other molecule we call a quencher, okay? But when the polymerase comes across the DNA, it actually chews this fluorescent molecule off, it gets away from its quencher, and it emits light, right? And so as the DNA is copying, more and more light gets emitted, right? And so a machine called the qPCR machine detects the light from the probe, and this is kind of what the readout looks like. So in this case, you're starting to detect light around cycle 20, and it gets exponentially, or it gets brighter as the cycle's time goes on, right? And so this is actually a pretty decent hit here at cycle 25. All right, this is very, very sensitive and very specific, right? So if the piece of DNA is in the reaction, we're probably going to find it. Okay, so back to this. So what we're looking for in this chip then is 33 nanoliter qPCR reactions. We have a specialized machine that can read these. So we're able to test 48 samples for 27 different pathogens, actually ×4 at a time. So we're generating about 5,000 data points every 2.5 hours or so. Okay. And the data that this makes kind of looks like we can plot this data like this. So this is the prevalence of different pathogens over time, and this is leading up to the pandemic. So you can see that in the winter of 2018, 2019, we had a flu A H1N1 outbreak before H3N2. In the summer, you kind of reduce all of those respiratory viruses, except rhino virus keeps hanging around. And then in the winter of 2020, we had a flu B, a very early flu B outbreak. So, and a shout out to Thomas Sibley, who developed this really awesome, incredible interactive data visualization. And you can find this actually at seattleflu.org/pathogens. And actually, when you roll over any of these pathogens, it lights up. It's really cool. And I recommend that you guys go see it. Okay. All right. So, and then the data gets to Mike Famulari, who is a mathematical modeler from the Institute of Disease Modeling, who developed detailed maps of where each virus was in the population. And this is a map of where the flu was on January 3rd, 2020. So this is Seattle, Vashon Island, about University of Washington's right about here. You can see that there was a lot more flu in South Seattle, and then kind of right around Lake Union for some reason at that point. And we were hoping that these kinds of really detailed maps could give people a warning that, you know, flu is coming. And back then we were like, oh, well, what if we could ask people to wear a mask? Nobody would wear a mask, they said. Well, here we are. Another thing we could do is get this, the data would go to Trevor Bedford at the Hutch, who is an expert in genomic epidemiology. And he would use the sequence viral genomes to understand how the virus was transmitting through the population. So how it was moving through the population and where it was going. And this requires the second quick digression, which is into phylogenetics, to make sense of what we're looking at here, this phylogenetic tree. So as pathogens replicate and spread, their genome needs to be replicated many times, right? So just like ours. Copying mistakes or mutations and accumulate in the genome. And these random mutations can be used to track the spread of the pathogen and learn about how it's transmitting and the dynamics of it, right? So maybe some of these are spreading faster than others. If we look at this cartoon viral sequence, we can see that they all share the same blue mutation. So that must be at the root of the phylogenetic tree for these 5 viruses, right? And all of the other viruses must have descended from it. Next, you can see the cyan or yellow mutations are in every sequence, but they're mutually exclusive. So we can draw 2 different branches for each of those viruses and their descendants, right? And so if we go back to this, this phylogenetic tree, it makes more sense, right? So what you're seeing is kind of 3 distinct different substrains of influenza, and then you can see that these 2 viruses are very closely related to each other. They probably only differ by a mutation or 2. And then because we had really detailed information of where all of the samples were coming from, we can see that the viruses were actually coming into North Seattle and spreading to South Seattle, which was kind of interesting. So this is the kind of information that you would want in the case of a pandemic, to kind of understand how viruses are moving through a population. So by late 2019, we had built what amounts to an unprecedented plumbing system, basically, for observing respiratory pathogens in a population. So we had linked up, we had built relationships with the population of Seattle, with schools and shelters and hospitals, and all of this was hooked up to advanced diagnostics and sequencing, which allowed for detailed analysis. And this kind of elaborate plumbing did not exist at this scale anywhere but here in January of 2020. So like everybody else, we were watching the news from China about an unexplained epidemic of pneumonia. And then the first genome was published on January 11. And this was really bad news, right? Because we understood that this was a novel coronavirus that looked a heck of a lot like SARS-CoV-1. And SARS-CoV-1, as we all know, had a case fatality rate of about 10%. So this was really worrying. There were also at the time no vaccines against the coronavirus. And interestingly enough, this is actually how the world learned about the genome of this virus. It was released on Twitter by Eddie Holmes, and who's a virologist from Australia, who collaborates with the Zhang lab in China. And the Zhang lab had actually sequenced it. And this actually links to a Word document, which is kind of crazy. Anyway, the next 12 genomes were also really bad news. So again, you guys now know how to read one of these phylogenetic trees. We're looking at the number of mutations here, and what you can see on this phylogenetic tree is that 1, 2, 3, 4, 5 of these viruses are exactly the same, from kind of random people sampled from the Wuhan outbreak. These 2 folks were actually from Thailand, and these people from China. You can see that 5 of these are the same. 2 more of them only have 1 more mutation. Another 4 only have 2 mutations away, and a third one has 3. So this is bad news, because this means that the virus is not continually spilling over from its animal host, like was suggested. This virus, it seemed, was spreading from human to human, and from what we could see on the news, it was spreading pretty quickly. So I can actually, our story was actually made into a comic by Ethan Sachs from NBC. And so if this virus was repeatedly jumping from humans to an animal reservoir, one would expect a lot more genetic diversity. And so, Trevor is really worried about this, and he tells me and Debbie actually and Kelly was there as well, that he was worried that it could be as bad as like the 1918 flu, meaning millions of people could die. And dinner, and you can kind of see I'm about to spill my soup here. So this is really bad news. But, and this is meant to be Helen Chu talking, she is saying that we could really make a difference with our lab capacity and all the samples that we had from people with respiratory illness in the area, right? So again, we had samples from hospitals, we had samples from the community, from all sorts of people who had respiratory symptoms. So, could it be in the community already? Remember, this was, mid to late January of 2020. We just really had to develop a test, right? So this is Helen saying, Lea, get on it. Okay. So from our PCR lesson earlier, everybody in this audience should be able to develop a passable test, right? All we need is the genome sequence of the virus, which we have. It was released on Twitter a few days before. And if you're, and you need the genomes of viruses that could be kind of similar, right? So we want to make sure that we're not finding the common cold seasonal coronaviruses if we're looking for SARS-CoV-2, which actually I don't even think was called that at that point. I didn't even end up designing this myself. I called experts at Thermo, who designed this stuff for a living, and they sent me some awesome test probe sets, which we actually still use today. They had no cross-reactivity with seasonal coronavirus. They detected molecules, SARS-CoV-2 molecules down into like the single digits, right? This is important because you want it to be really sensitive. And then the specificity was really important early on too, right? Because like we didn't want people in spacesuits showing up on somebody's lawn because they had a cold, right? That would be bad. So we developed this test in 384 well plates with 190 samples in duplicate, which was, a pretty high throughput test at the time. The CDC had developed one that was 22 samples at a time, and we certainly weren't going to use that. Okay. All right. So I had developed this test and shown it was pretty good by February 7th. But during this interval, it had become clear that the CDC had also developed not only a slow test, but a shitty one. They had sent public health labs around the country a test that was contaminated with a control piece of DNA. Then they required that they were the only lab that could do the test, but the persons of interest or the people who they're sending the test to had to have direct travel history to China, never mind the massive outbreak in Europe. And then there were also a lot of other ethical issues to figure out, like what do we do if we find a positive case? But it was getting more and more obvious and dire to our modeling teams that COVID was out there, right? They knew how many people had gotten on a plane, and they were like, it's here. So we finally managed to get tacit agreement from the CDC and the state that we could test these samples coming through the lab, but with the caveat that we weren't supposed to identify anyone who the person who the sample came from. So we were supposed to, kind of talk to the state and be like, one case of SARS-CoV-2, But anyway, it was kind of ridiculous, but here we go. So we started testing the samples that were coming through the lab on February 25th. It only took us 2 days to find a positive, right? So as you guys know, this is what a qPCR result looks like. As the cycles go higher, you get more and more signal from that copying DNA. And this was very, very positive. This is supposed to be a picture of me running back to Faggy, running back between Genome Sciences and the building I work in. We had just had a meeting talking about like what we would do if we found a positive, and I was just trying to catch everybody before they took off. So I reconfirmed the positive that night, because again, we're really worried that, maybe we got it wrong, but I used a different test this time, one that I had bought from another company. And the next morning, I drove it to the public health lab up in Shoreline. This is the box in my car, and this is the cartoon version of that. And we knew and they also confirmed that it was indeed SARS-CoV-2, and we'd also sequenced it, and that showed that it was definitely SARS-CoV-2 as well. And we knew the sample had come from a teenager north of Seattle. We had known that it had come from the Seattle Children's Clinic days before. And although we weren't supposed to, we felt that we had an ethical obligation to inform the patient, right? So it turned out that this person had a really high viral load. And we, as doctors, our clinical team was worried about this person, and also the entire Seattle Children's Clinic had basically been exposed as well, right? So they were going to have to quarantine. And so we actually, so this was, it turned out to be a high school student from north of Seattle, and the mom managed to, he was feeling better that morning, and the mom managed to get a hold of him just as he was pulling into school after being out sick, and told him to come home. So, thus avoided possibly kind of exposing the entire high school. And, this is what really kicked off the public health response in and around Seattle. The tech companies started to close. This was not, this was not a travel case. This kid had no connection at all whatsoever to travel. And then, just right after this was the long-term care facility up in Kirkland, where unfortunately some of their patients started to become very ill. So from our samples that were coming through our hospital and Swab and Sense, we kind of kept testing these samples at this point. And remember, we were the only people in the United States that were testing people who hadn't traveled and weren't dying of pneumonia, right? So we were the only community samples in the US. And so we were one of the few labs in the country who were able to kind of estimate the size of the outbreak before we could even start testing for it, right? And so we were able to do this by two ways. One was phylogenetics. So again, all of these samples were kind of sequenced by us and by our neighboring lab, the University of Washington Virology Lab. And you can see that a lot of them are very similar. And so because we know how fast SARS-CoV-2 mutates, it gets one mutation about every 3 weeks, we can tell by the diversity of these genomes, basically how long it had been in the population before we started testing for it. And then again, using very different methods, Mike Familare was able to estimate just from the population, knowing the denominator of the number of tests we did and kind of where those samples came from, how many people they thought, and they kind of coalesced around that the outbreak in Washington was already at 0.05% of the population, or a few thousand people by the time we started looking. But you'll notice here that all of these plots end at about March 9th, because that's when the state shut us down. So we were not technically what we were called clinically certified. So we were not technically allowed to test samples for a communicable or a reportable disease like SARS-CoV-2. So we spent only, actually, this is like a minor miracle, only about 10 days getting our ducks in a row and jumping through hoops to be able to relaunch our testing program. And in the meantime, we had formed a partnership with Public Health Seattle King County, and we formed the Seattle Coronavirus Assessment Network. So about, I think we were shut down from March 9th to about March 23rd. And so what SCAN was, or the Seattle Coronavirus Assessment Network, was the very first community surveillance program for COVID. And since we launched in March 23rd, 2020, we've delivered and tested 56,000 kits and discovered about 2,000 positive samples. And you can see the kind of samples through time. We had a big spike right at the beginning. I'll go through what this is in a second. and then kind of like waning interest in the spring here. You can see this is just a dashboard. You can see this on scanpublichealth.org. You can see kind of where the samples came from and where the positives came from. And then you can see the positivity rate over time. And you can see that, you know, it's kind of low, and you actually see it really get high here. And kind of during the pandemic, we've kind of become, as the pandemic's gone on, we've kind of become a contact tracing arm of Public Health Seattle King County. So the samples we're getting now are just very much more likely to be positive. But in the beginning, again, we were the only community testing program, and we were, you know, I think we had done a fairly decent job of passing out the sample, the kits kind of randomly. So we were able to get, you know, really nice community estimates of, you know, the estimates of the prevalence of SARS-CoV-2 through this program. And this was really important because I don't know if you guys, I'm sure you guys remember that testing was really hard to come by in spring of 2020. So we were doing about 25% of all COVID testing in Seattle, even with only just our few hundred kits a day over that time. Okay, and all of this data, of course, was going back to Public Health Seattle King County and the state in order to help them make better decisions. So what happened here, right? So obviously we stopped testing for a while, and you'll notice a little pause. So the FDA actually had revised their guidance around at-home sample collection. And although we had submitted our paperwork for an emergency use authorization, now that everybody on the planet knows what emergency use authorization means, we would no longer be able to test while it was pending, right? And this was because of the at-home component of our test, among other things. They were worried that, you know, the usability of the test and the validity of the data for swabs that were collected at home and unsupervised. So at this point, we had kind of scaled up, and I've got a whole bunch of technicians who are really smart and they're bored. And so they thought that they would kind of use our data set to see if we could figure out just how much mistakes made by participants really affected the results of the tests. And, you know, was this really the big deal the FDA said it was? And, you know, that caused us to get shut down again, right? And this was, you know, a really unfortunate time to be shut down as well. Okay, so this is a Midturbinate swab. It goes about, it's not one of like the brain scrapers like those nasopharyngeal swabs, nor is it kind of a Q-tip. Again, it goes about halfway. So the stopper stops you from shoving it into your brain. And then you swab yourself, and then you put it in the transport media and break off the handle, right, so that it fits in the tube. So this part goes in the tube, you're supposed to throw out the handle. Well, you would think that would be pretty easy, but 1% of the tubes were returned with the handle in the transport media instead of the swab, suggesting that people might have used the swab backwards, swabbing with this hard plastic handle instead of this like nice flocked pointy swab. And so I told you earlier, you guys just learned about PCR and learned how sensitive it is. You know, could PCR-based testing still detect pathogens? Okay, so we tried to compare the two, right? So one of the things we test for is human markers, right? We want to make sure that people actually shove these swabs up their nose. Sometimes they don't. And so you can see that, you know, the swabs versus the handles had a pretty similar rate of failure for lack of human RNA. So 2.9% for the handles versus the swabs, but it's insignificant. There was a little bit more of the human marker. It's called RNase P on the swabs versus the handles, but the pathogen detection rate was indistinguishable. We found all sorts of things on these handles, from SARS-CoV-2 to influenza to rhinovirus. This is actually published. And who did it, right? We wanted to kind of know who were the ones putting the swabs up their nose backwards, because the beauty of the Seattle flu study was we had all of this really detailed demographic data. And it turns out that they were the over 50 set, significantly. And, you know, and the funny thing was that they actually felt more confident that they had swabbed themselves correctly And they had less discomfort, which makes sense because they used the blunt end rather than the pointy end. So basically what all of this data shows is that you can screw up swabbing pretty badly and still get, a valid result. And, the FDA was kind of being ridiculous, but whatever, water under the bridge now. So we figured out our regulatory situation and we were back in business, although to this day, we are still not FDA approved, and we can get into that later if you want. Okay, so again, we had figured out our regulatory situation, and we were kind of able to scale up and take on a pretty big project, the Husky coronavirus testing program. So this was to kind of help the University of Washington open up more safely. So we had already switched to these lovely Q-tip adjacent swabs because we couldn't get those pointy ones, right? This was the supply chain problem of July, kind of after we started from the FDA pause, was that we couldn't get the swabs anymore. And what we had realized early on was that we kept having massive supply chain problems that exist to this day. And so right after the Husky coronavirus testing launched, my lab manager actually got a phone call from the maker of this machine, Roche, big French company, Swiss maybe, Swiss. And he had just put in an order for more extraction reagents, right? So I told you before, we have to crack open all of these viruses to be able to detect them. And they were telling him that we didn't rank high enough, we weren't a big enough customer, and they would no longer be supplying us with extraction reagents. And so now this stupid thing is $140,000 paperweight. And we have samples coming from SCAN, they're coming from homeless shelters, and now they're coming from the University of Washington. Kind of what were we going to do about it? So early on, we had asked, do we really need to extract the RNA from these samples? Again, I told you that PCR is really, really sensitive. Can we get away with not doing that? And this was helped here by the fact that we had already switched to these easier to deal with swabs, and we could elute these maybe in a PCR-friendly buffer. So could we develop an extraction-free protocol for nasal swabs? And it turns out we could, which was really exciting. And this was led by actually a graduate student that I stole from Genome Sciences, Sanjay Srivatsan, and a lot of the rest of the many others in the flu lab, and Beth Martin and Tina Lockwood, who's our clinical director. So again, they were using this anterior swab. We were transporting it dry. So the dogma for many, many, many years, and a lot of people still do this, they transport it in this buffer that became really hard to get. And so we were transporting them dry, just in an empty tube, which was great. So the more things we could strip out of this testing process, the better. We eluted them in a really easy to, we kind of rehydrated them and got the stuff off in a buffer called TE. It's a really simple buffer. Any lab in the United States could make liters of it for, $50. And then we just basically put the samples in a plate. We added something that digested snot, basically. Some of the snottier samples were causing us problems. And then we just heat them. So one of the things you're worried about, and one of the reasons you extract the RNA, is to kill the virus, right? So we wanted to make sure we were killing the virus and not spreading it all over the lab. So we just heated the crap out of them to basically almost boiled them for about 15 minutes, which meant that the virus wasn't going to survive. And then we can put those right into PCR reactions and detect the virus. So the per sample cost, right? So for extraction, it costs about $5 a sample. And then you have to do it sequentially instead of all at the same time. So we saved a ton of money by moving to this extraction-free protocol and a lot of time. And this was, again, we were able to scale up and actually share this protocol with others, which was really nice, or really important thing to do. So, did any of this work, right? And that's kind of hard to quantify sometimes, but the Husky testing program did work. So if you look at this plot here, this blue line is the average positivity average positivity of King County on the whole, whereas the other lines, the orange ones, are just the Greek students. You can see we had a big spike with them right in the beginning, and then right after Halloween, of course, and then right after Thanksgiving, and then the rest of the university, so employees and non-Greek students were way lower. And then by the phylogenetics, again, you guys know how to look at these now. You can see that the viruses within the university were kind of bouncing around within the university and not spreading out into the community, right? So you're not seeing a whole bunch of community cases spread off from this cluster, right? So this is suggesting that Helen and her team and the rest of us were able to kind of keep and you know, lots of work went into this by a lot of people, including the Environmental Health and Safety Program at the University of Washington, to kind of keep this virus within, from escaping the university. Okay. Also, this genomic data is reported directly to the Washington Department of Health in Seattle, King County. This is Cassia Wagner. She's a badass grad student in genome sciences. She works for Trevor. And what this is actually data that we showed the Department of Health a week and a half ago. So we give them situation reports. This is showing, I should have put the new names on here. This is Alpha, B117, and you can see that that's kind of tailing off and actually starting to decrease in frequency. But what we can see is that this is Delta down here in purple, and you can see that Delta is kind of on the rise and pushing out Alpha or B117. And so we're able to kind of really share information with them almost in real time, which is really important. So the death rate, so even though, you know, Washington was an early hotspot, right? So likely, possibly because we looked. And remember, early detection is important when battling exponential growth, right? And we recorded the first 50, Washington recorded the first 50 deaths from COVID, but since then we've been crushing the curve in comparison, right? So kind of ignore the colors on this. This is stolen from the Johns Hopkins Coronavirus Research Center, but I just wanted to show the epidemiological curves from all the states together. And you can see that Washington has actually been kind of flat almost the whole time in comparison to places like North Dakota, South Dakota, Wyoming, basically, most of our neighbors except for Oregon. So we rank like 46, I think, in cases per capita, 44th in deaths. And that adds up, and then if you compare our death rate to the rest of the United States, it adds up to about 400,000 people that might still be alive if the United States had the same death rate as Washington State, right? And so, you know, why was that? You know, Washington had a lot of really great scientific resources, and, you know, possibly a population that was more willing to listen than others. So what did we learn kind of this whole time, right? So that would be important for the next pandemic, right? So we learned that people can handle self-swabbing, right? And this is kind of important. One of the dreams is that, you know, kind of everybody has a swab on hand, and they get sick, they swab it and they mail it in, right? You know what everybody has kind of all the time. We learned that community engagement in that same vein is really valuable. We learned that the US lab regulatory landscape is a mess. And to be quite honest with you, this would happen exactly the same way if this were to happen again. Preparation and flexibility is a key. And again, you know, being an academic lab, we were able to kind of flex around the regulatory mess and also recurrent supply chain problems, right? This was not something, you know, we work in the 20th century. When we order a piece of plastic, we expect it to show up. You know, we're still having, and now this is spilled over to biotechnology in general, and basically, and you know, general consumers. But when everybody's trying to do the same test at the same time, supply chain's going to collapse, right? And so being able to be flexible was key. We also learned that actually some of this was very, very expensive, right? I told you I was able to develop this like stupid cheap test, right? But actually getting the samples cost an arm and a leg. So these are kind of, you know, a lot of the things we learned, and what is the vision for the future, right? And where does that take us in the case that this was going to happen again, right? And what we would hope to build, or what we would hope the United States would fund, would be the Seattle flu study on steroids, right? So Seattle flu study, it was actually not a government funded project. This was funded by actually Bill Gates, Gates Ventures, so his private office, not even the foundation. So this was kind of a vision of his. And so what we would want for the future, right, are funding mechanisms to make pathogen surveillance systems sustainable, right? So right now it's not, right? We spend extraordinarily, well, you know, I want to say we're spending a lot of money, but in comparison to kind of what happened and you know, preventing it from happening again, you know, it pales in comparison. So, you know, can we have a mechanism in place that is, you know, kind of stably funded to make like a respiratory disease observatories, right? And this would require political buy-in from local, state, and federal governments to maintain these sorts of platforms. And then again, I just told you one of the reasons we need to do this is because it's expensive. Can we explore new models to really minimize the cost at acquiring the samples? And both of these things would kind of require changes, major changes to the regulatory processes. One of the reasons it's very expensive is because there's certain things we're just kind of not allowed to do for quasi-stupid reasons. We have to employ people who just talk to regulatory agencies. They're awesome, don't get me wrong, but like if it would be a cheaper thing to do if that didn't exist. We would encourage people, or we would encourage, you know, Seattle Flu Study Part 2 to build pre-existing relationships with public health entities. So for example, back in March of 2020, part of, you know, even during the shutdown, part of what we were doing was negotiating with Public Health Seattle King County and, you know, filling out memorandums of understanding and all this kind of crap. You know, we were certified 2 days before we started testing again because they were working on like their branding, and I was like, you got to be kidding me, right? So like, can we kind of have this kind of stuff set up beforehand so we don't have to deal with that kind of, I'll use our new president's word, malarkey. Again, stockpiling testing reagents, right? Again, anything with a genome, we know the ingredients to a test, right? Some of this stuff expires, but again, you know, the manufacturing capacity really needs to be there. And then again, a national framework to harness the power of the United States science community. The US is a science powerhouse, right? But somehow, you know, we were the ones that fell flat on our faces, whereas, you know, places like Korea and England were able to kind of harness the power of their academic labs, where the academic labs here were kind of more shut out because we kind of focus on empowering the bigger manufacturers. But, you know, again, that was important as well, but, you know, shutting out the academic labs really kind of shut things down, especially early on. So I'd like to thank my team. So this is the flu team, the Seattle Flu PIs. We have funding again from Gates Ventures. I'm now funded by the CDC to do some of the sequencing. And then the Northwest Genome Center, right? They do a lot of the sequencing. They've been instrumental in getting, actually, Machiko actually is the cat herder in chief, getting a lot of these samples from around the state. Acknowledgements, again, we work with many people, including Seattle Public Health, Public Health, Seattle King County, Washington Department of Health, while they were a barrier in the beginning, have become really great at allowing us to stay flexible around some of these supply chain problems. And they've been a really great partner since then. The FDA, again, we still don't have an EUA. And again, this is just most of the people who have worked on this project. It's a huge, huge undertaking. It's a huge team. So again, when we talk about making this sustainable, you're looking at data and infrastructure that took hundreds of people to put together. And so to kind of replicate this around the country, this is kind of what it takes. So I will stop there, and anybody can kind of feel free to ask questions.
Yeah, I see there's a question from David Gordon.
Okay, so here, let me sort of give you the background of this question. So when people say cases are increasing, or people say cases are decreasing, I totally reject that, because I think that's only a function of people who are worried and therefore get themselves tested. And it's a very tiny fraction of the total number of people that are really infected, most of which don't get themselves tested. And so even to say the trend, I don't trust that. But what I would trust, and they were doing this a little bit at the beginning, was compulsory random testing. And this was, I think, of automobile accident victims who were admitted at the hospital. They were all tested. So in doing that, if you saw an increase in the fraction that tested positive, then you could really much more reliably conclude that the amount of COVID within the community was increasing, and vice versa. And so I would really like to see more of that done. Do you know anything about that?
Well, there's a decent amount of random testing, right? Husky testing, for one. We called people, I don't know if you had signed up, David, but that we were calling people in kind of randomly. So that's a ton of asymptomatic random testing right there. We're testing hundreds of people a day. And then there's a lot of other sources of random testing. So people going in for surgeries at the hospital, any hospital admission right now for unrelated things related or unrelated to COVID, people are getting tested. There's a lot of random testing for workplace and things like that. So there's not random testing going on. And in addition, we have orthogonal ways to tell whether or not the case count is rising or falling, and that's actually through the phylogenetics and the transmission dynamics, right? So, Trevor is able to really, and the percentage of cases that are actually being sequenced in the United States has increased dramatically, to the point where in Washington we're sequencing 20% of the positives, which is an incredible thing.
Where do you see the data? Where could I go online to see the data on the random tests?
It's just all it's all kind of mixed into the line list for the Washington State.
Okay, so it's mixed into all the people that thought they were sick, they were worried, so they got tested.
Yeah.
Okay, so I can't really see the percentage of just the random then.
No, but the Department of Health should, I think the Department of Health may or may not, I don't know if they know whether or not why people were tested unless they ask. But I mean, I think all of that goes into the modeling that, the state and other epidemiologists use to try to understand whether or not case counts are rising or falling. And obviously, we know that death rates are pretty stable, about 1%.
Yeah, I completely go by the number of COVID deaths. That's the only thing I do trust.
Well, yeah, so it lags about 2 weeks. So you can tell where we were 2 weeks ago about the death rate now. So
thanks.
Yep.
So I'll try to alternate raised hands and the chat. Celeste, would you like to ask your question?
Celeste, you're muted if you're trying to talk.
Maybe while we're waiting, would Venkatesh like to ask their question?
Sure, thanks. If you could take one thing from the relationship between King County and the Seattle flu study and replicate it to another city to notice future pandemics early, what would it be?
Man. That's a really good question. You know, that's a really, really good question. They've been a little bit of a silent partner of ours. I think they've been very, very busy and, you know, until recently badly underfunded. So I have to say, we did a lot of the work, but just having their name associated with us kind of made us a trusted partner. So just actually having that partnership kind of allowed us to be kind of piggyback off the trustworthiness of them. And Seattle Public Health King County is actually a very, very good public health entity. And I think people trust them. And so I think that kind of bled into trust for us as well. So we were able to kind of launch and you know, have people understand that, you know, this was a trustworthy endeavor and like you weren't kind of sending your test off into the ether for, you know, a non-result. So that kind of like lent credence to the program for sure. You know, who are these randos from University of Washington?
Thanks.
Yeah.
Hey, Leah, sorry.
What?
I'm on my iPad and I couldn't unmute. Can you hear me?
Yeah.
Yeah, I had put my question in the chat, but I'll just ask it. First off, awesome, fantastic work, great talk. But a question regarding the next virus, the next novel virus that comes out. Your test requires that you actually know the genome of that sequence. So you would presumably miss something really new. What would be the process for trying to identify that new virus? Would you have some patient that was really ill but didn't show any positive on all your tests? So how would you go forward?
Well, multi-pronged. So first of all, so again, we were expecting this to be influenza. And we have part of one of our qPCR, there's parts of influenza that really don't change. So we do have, you know, tests for, you know, specifically H1N1, and then we kind of have like pan flu A, pan flu. So, you know, had it been, you know, more similar to, you know, influenza, which, you know, we would have picked up, that up. Another thing that we kind of, you know, one of the things we want to do, but there's always been some other fire to put out, is build a metagenomic sequencing platform.
Oh, that would be great.
Yeah. So again, because we have all of this amazing data, we've got, you know, samples from people who were clearly sick in the hospital, possibly even with pneumonia, and then they were negative for all 27 pathogens. So what did they have? And can we find it? And so we had kind of always been planning on building that when, you know, we had to put other fires out first, but that is definitely on the list. And also the sequencing we were doing was done by hybrid capture. So kind of like exome sequencing.
Mm-hmm. Yep.
And it doesn't, you don't have to have it perfect. Like for example, we had SARS-CoV-1 probe set in there.
Yeah. Mm-hmm.
I always want to do the experiment. I don't know if it would have worked. Like I guarantee, I really think that those probes would have picked up SARS-CoV-2. as well.
But would it would have been, or I guess you would have known it was at least this type of virus, right? Okay.
Exactly.
Yeah.
So yeah, what was this? You know, I don't know. So again, we think with the hybrid capture that, you know, we would, because, you know, you can get up to about decent capture with about 80% identity homology.
Oh, that's really good. Okay, great. Thanks.
Yeah.
Ellen Weissman, would you like to ask your question?
Can you hear me?
Mm-hmm.
You can hear me. I'm on a different device, so I'm not sure if you could hear me. I just have one comment and then I have a real question. I find I'll just read what I said because, I've just been floored by this new approval of the Alzheimer's drug. I'll put that in quotes because it's just a travesty that was approved. So here we have you've gone a whole year, actually more than a year, without FDA approval, and clearly you're doing a great job, doing fantastic things, and you don't have to approval. And we have to fight this stupid approval that the FDA has now put out on a drug that shouldn't be approved. So, it's just sort of my kind of jaw dropping, if you could imagine it. It's just a comment that there's something wrong with the regulatory system if both of these things can happen more or less simultaneously. One that both shouldn't have happened. Anyway, it's a comment. So now I'll just ask a separate question. So you commented on the Seattle public health people, and they're very, very good. They're really outstanding. So I want to ask, and just sort of, just you think a little. We have really good public health people, and we have really good genomic people, genomicists here. Mm-hmm. How much of what you were able to do, given what happened, is really just this fortuitous juxtaposition of two absolutely outstanding groups that have complementary skills and are very good at what they're doing? I mean, you were sort of saying it would be nice to have more, public health, more surveillance and so on, but how much of this would only be able to happen here or maybe in Maryland or, a few places in the country that have kind of similar quality of people in, 2 or 3 areas that you need to pull this off? I mean, I think we also have to say that the School of Medicine has done an absolutely outstanding job also at preparing and keeping cases down too. I mean, that didn't even come into your story, but that's a third leg of this whole story. So, what are your thoughts?
Yeah, and the Broad did the same thing. They were doing 100,000 tests a day, right?
Right.
Able to scale up. So again, that goes into tapping allowing the regulatory agencies to be flexible enough to tap the power of the academic science, right? So again, that does put other places at a disadvantage. So, places without
South Dakota.
Yes, they've got bigger fish to fry than lack of neck seeks. But so they were also, I think, fighting political headwinds there. But one of the things that the CDC has done recently is poured a ton of money into local public health agencies. So for example, Debbie's lab, I think Debbie was here, actually trained our local Department of Health to do COVID-seq. And I think we're going to teach them how to do hybrid capture next, so that, they're now, from us, they're like, oh, well, maybe we should sequence all these viruses. And so they don't have to change, the way we do COVID sequencing now is just amplicon sequencing. So for each virus, you would have to have a bespoke assay. But with hybrid capture, which is like an exome, but for all viruses, you could use the same probe sets and do the same sequencing, right? And so, so that they don't have to kind of switch back and forth depending on target, can we teach them how to do that? So Debbie has been instrumental in actually kind of spreading our academic sequencing knowledge to our local public health entity. And hopefully, Wyoming's public health lab, actually, they've got the highest percentage of SARS-CoV-2 genome sequenced of their positive cases, because the Wyoming Public Health Agency has been kicking ass sequencing.
Interesting. Yeah. So. Thanks.
Yeah.
Maitreya, would you like to ask your question?
Yeah, sure. I was just curious if they ever figured out how that first teenager that you guys discovered got exposed.
Nope.
Was contact tracing helpful? They never figured it out. Wow.
I really don't think they ever figured it out. And how far back it was before there was a travel associated case, but no. And actually, Trevor and they estimate that it had been kind of in the area for about 6 weeks. So that kind of kills your ability to do great contact tracing, right? Because we weren't allowed to test anybody who wasn't from China or hadn't had direct contact with China at the time. So, all of these people who may or may not have been very, very sick, there were people with pneumonia at Northwest Hospital, they couldn't get tested. So they still don't know. I should ask Jeff Duchin next time I talk to him though.
Well, thanks, Leah, for that really fascinating talk. We can let everyone go to grab your own post-talk drink, but we're going to have these every Wednesday in July, so hope to see you back for the next one.